Okay, hi everyone. Today we're going to talk about control of microbes and control of microorganisms in the body. So you may remember the last two chapters we talked about controlling microorganisms and we talked about physical and then chemical control, but both of those were controlling microorganisms in the environment. This chapter now focuses on antimicrobial treatment. So that's control of microorganisms inside the body, say inside a patient. And we're going to look at various aspects of that. Okay, so to get started, let's talk about vaccines. A vaccine, we need to define our terms. A vaccine is a weakened or dead form of a pathogen, a disease-causing organism. Vaccines should not cause disease in most hosts. There are exceptions. People may have an allergic reaction, for example. But vaccines should not cause disease in most hosts, or maybe only a mild case, because they're a weakened or dead form of that pathogen. However, even though they don't cause disease in most hosts, or a mild case, your immune system doesn't know that. So when you're given the vaccine, your immune system thinks it's a full-blown invasion by that pathogen. So your immune system responds normally. So your immune system kicks into full gear, fights this thing off, and that helps you build immunity if you're ever infected by that same pathogen again. Little history, Edward Jenner, pictured here, was a British doctor who developed the first vaccine way back in 1796. It was against a dreaded disease called smallpox. Smallpox now has been eradicated from the human race. So smallpox, people don't get smallpox anymore. Smallpox was eradicated back in by about 1980, was 1977 was the last natural case. But up until that point, smallpox was a very dreaded disease. Earlier work before Jenner had been done in ancient China and in the Middle East, and a woman named Lady Mary Montagu did work in about 1717 or 1720s in England, predating Jenner. So scientists always build on the work of others. This is what smallpox looked like. And one of the really bad things about it was even if you survived smallpox, which people did, it would leave these nasty scars on you pretty much for life. Look at the effectiveness of vaccines since their discovery by Edward Jenner. So over here is the pre-vaccine era estimated annual morbidity in the United States, so number of cases in the U.S., and then the most recent reports after the vaccine. So 100% decline in diphtheria, 99% decrease, and you can see all these here. Absolutely incredible tool for combating infection. So like this says here, got polio, and then there's the asterisk. All these other things mean neither, thanks science. This child says, what's that mark on your arm, Mama? My smallpox vaccine scar. Why don't I have one? Because it worked. All right. So how do vaccines work? When you are first given a vaccine, like I said, that kicks in your immune system. This is called the primary immune response, or titer. So this refers to the first exposure to a vaccine. The secondary immune response is the second exposure to the vaccine, and this would also work if you contracted a disease naturally. So the first time you get a disease, you would trigger a primary immune response, then the second time is a secondary immune response. Now, so you can see here, this y-axis is tracking the relative concentration of antibody in the system. So what happens is if you get a disease or are given a vaccine, your immune system kicks into gear, and one of the things your immune system makes are little proteins called antibodies. We talked about these a little bit in a previous chapter. antibodies can bind, lock, and key to what's called an antigen, which is the part or piece of the disease-causing organism, the pathogen, that's triggering the immune system. So we notice down here, when you get this the first time, over time, antibodies build up in the body, and then once the infection starts to wane, this drops down. This is your primary response. But look, a whole bunch of time could go by here, weeks, months, even years. If you're infected by that same antigen again, look how quickly the body produces antibodies against it. It's just an overwhelming response. And the reason your body can do this is because during the primary response, your body makes what are called memory cells. Memory cells create what we call immunologic memory. In other words, your immune system remembers what it has fought off before, what the body has been infected with before. And it can react very quickly to any antigens it's been exposed to before. Very, very quickly, as this graph illustrates. When you get a booster shot, you're sort of speeding up that secondary immune response and keeping those antibody levels high. All right? Now, what are different types of vaccines? There are several. They're made in different ways, and each one has advantages and disadvantages. There's always trade-offs with these things. So, number one is what's called an attenuated vaccine or a modified live. This is a weaker, less pathogenic version of a live organism. So it's still a living thing, okay, but it's a weakened version of it. These remain active in the body, so you usually don't need boosters. Number two, you could have an inactivated whole agent. This is a whole pathogen, the whole disease-causing organism that's been killed. We put it into a vaccine. Boosters are often needed. You could use subunits. Subunits are only pieces of a pathogen. Again, boosters often needed. Toxoids is where we make a vaccine not from the pathogen itself, but from the toxins or poisons or substances that it produces. These are pretty long-lasting, but again, boosters often needed. Conjugate vaccines is where we take a strong protein, the antigen, and we chemically bond it to a weaker polysaccharide. Both the polysaccharide and the protein can trigger an immune response. So they are longer lasting but expensive. We can use DNA fragments. We can use recombinant DNA that was made in the lab, different pieces put together. Most recently, for like the COVID vaccine, they used messenger RNA. This is where part of the viral genetic code is injected into the body. The body makes these viral proteins, but not the whole virus. And the immune system makes antibodies and other cells called T cells to fight that virus. Now, this diagram here shows some of the different types of vaccines, a description, advantages, disadvantages, and examples of diseases that use them. Okay? Here are again principal vaccines to prevent human disease from recommended by the CDC. All right, you can see the vaccine type, how it's administered, and so on. This is how that COVID vaccine was made using that messenger RNA technology. So you could take a look at that if you like. These things that look like capital letter Y's represent antibody. All right, now you may have heard when it comes to vaccines or spread of disease, what's called herd immunity. Herd immunity is where a large percentage of a population becomes immune to an infection. And they become immune either by getting a disease and naturally recovering from it, or through vaccination. Either way, a large percentage of the population becomes immune to infection. When this happens, everyone, it provides protection for individuals who are not immune. Say those who are immunocompromised, or organ transplant patients on immunosuppressive drugs, or young children or newborns, or someone else who just may be predisposed or genetically predisposed to some kind of infection. So if a large percent of the population is immune to the infection, it provides protection for those who are not. So as more and more people become immune, say through a vaccine, this disrupts the chain of infection, which in turn stops or slows the spread of disease, meaning that those who cannot be vaccinated, or those who are vulnerable, not immune, have a less chance of being infected. Now, everyone, for this to work, you have to reach what's called the HIT, or herd immunity threshold. This is the percent of the population that must be immune for this protection to work. Okay? Once we reach this HIT, the disease is gradually eliminated from the population. Okay? It becomes less and less common. Now, this will only work for contagious infectious diseases, not all infectious diseases, only those that are spread easily. So in this diagram here, blue is a person who's not, say, vaccinated, but still healthy. Yellow is vaccinated and healthy, and red is sick, not vaccinated, sick, and contagious. So in the top panel, we see here no one is immunized. All right? A few people get the disease in red, and it spreads because it's contagious. It spreads rapidly. And my whole population is now almost infected. In panel 2, some individuals are vaccinated. So these are the ones in yellow. So here, these two individuals in red get sick. With some of the populations immunized, the contagious disease spreads, but not as much. It still spreads, but a little bit less people are infected. Now, why haven't more been protected? We haven't reached that herd immunity threshold. Now, that's what shows it down here. If most of the population gets immunized, so see, most of them are yellow, all right? A few in blue are not. And then the red ones get sick, the spread of the contagious disease is contained, okay? So you'll notice here only one other person got sick, all right? So in the bottom box, a large proportion of the population have been vaccinated. This prevents the illness from spreading significantly, including to those who are not immunized or vulnerable. Now, this chart here shows the HIT for different diseases. So here's your disease, how it's transmitted, and what percent of the population needs to be vaccinated for herd immunity to occur. So it can be quite high. All right? All right, now let's shift from vaccines to drugs. Remember, we're talking about antimicrobial treatment in the body. Now, antimicrobial drugs are drugs that work inside the body. Now, Alexander Fleming, pictured here, first discovered penicillin in 1929. Penicillin is the very first antibiotic. He was working with a bacteria called Staphylococcus epidermidis, pictured here, and he noticed that around, this is the penicillium colony, this is a fungus. Around that colony, there's a relatively clear area where not a lot of bacteria are growing. And he realized that they're not growing there because this fungus, this penicillium, is secreting some kind of chemical, which we now call an antibiotic, that is inhibiting their growth. It's slowing them down or killing them. Penicillin came into heavy use starting in the 1940s, particularly after World War II. Later, this man here, Selman Waxman, discovers over 20 additional antibiotics while working with soil bacteria, and he would actually come up with the term antibiotic. Now, this man, a German scientist named Paul Ehrlich, discovers a cure for syphilis in 1910. This drug he names Salvarsan. Salvarsan. It's the first drug, not an antibiotic, but the first drug. He referred to it as a magic bullet. Now, what he meant by that was an antimicrobial compound that can target a specific species or related group of microorganisms while not hurting the host. Before Ehrlich's discovery, it was thought in the medical community that you couldn't specifically target, let's say, bacteria that are infecting someone. The idea was that anything that could kill the bacteria or that would kill the bacteria would also harm or kill the patient. It would kill their cells. Salvarsan was able to show that that's not the case. You could develop a compound, a drug, that's specific just to the microorganism. Salvarsan was initially a powder that had to be mixed with water. He is considered the father of drug therapy. And since his discovery, of course, thousands of different drugs have been invented and made and discovered, and it also ushered in the whole era of the pharmaceutical industry, which you may have opinions about. Now, what are some types of antimicrobial drugs? Well, first we have antibiotics, which I just mentioned. Now, an antibiotic is an antimicrobial that ultimately comes from a natural origin. Today, many can also be made in the laboratory synthetically, but initially and ultimately it comes from a natural origin. Of particular importance here, everyone, is the bacterial genus Streptomyces. This bacterial genus, this group of bacteria, produces more than half of all known antibiotics. Interestingly, Streptomyces, this is a soil bacteria. It lives in the soil. It also produces a compound called geosmin, which is what gives soil its kind of earthy odor, like that odor of, say, damp dirt or topsoil. That's coming from this geosmin here. Antibiotics, again, ultimately come from a natural origin. Today, you can also have semi-synthetic antibiotics. These are chemically altered antibiotics. Now, why would you chemically alter one that came from a natural origin? Well, semi-synthetic have several advantages over natural antibiotics. They can be more effective, longer lasting, easier to administer, and even cheaper. So, for example, penicillin that comes from a natural origin. It comes from that bacteria or that fungus that Fleming first saw on that plate, penicillium. We can chemically alter that to make amoxicillin, which is semi-synthetic. Okay? You can also have synthetic antimicrobial. These are completely synthesized in the lab. So completely made in the lab. Now, besides antibiotics, there are drugs, not antibiotics, but drugs, other drugs, can also be semi-synthetic or synthetic. Completely made in the lab or something that started natural and is chemically tweaked. We have antifungals, antivirals, and even antiprotozoans. So if we look at this chart here, this is sources of some common antibiotics and semi-synthetics. So we get many of these from fungi. Here's different fungal species, antibiotics we get from them. Bacteria, particularly look at all the streptomyces here, okay? And the different antibiotics we make from them, okay? This is streptomyces growing on an agar plate. This genus, for example, produces streptomycin, tetracycline, chloramphenicol, ivermectin, and many more. More than half of all known antibiotics are naturally produced and come ultimately from this genus, these different species in that genus of bacteria. So why do some microorganisms produce antibiotics? Like those fungi, like penicillium, it makes antibiotics normally. So do all those bacteria. Why do they do that? You might recall from a previous chapter, the chapter on microbial metabolism. Antibiotics are what we call secondary metabolites. These are complex organic molecules that, again, are produced along the path from reactants to products. So you might recall, if we want to make compound E, from our starting material, our reactant A, we have to first make B, C, and D. B, C, and D are secondary metabolites. These secondary metabolites are not essential for normal growth and reproduction, and producing them is complex because it's along a whole metabolic pathway. Tetracycline has 78 steps, each with its own enzyme. Erythromycin, 28 steps. So what do, now remember, these antibiotics that are made by, say, bacteria are not essential for normal growth and function, but what do they do in nature? First thing you have to know is when bacteria or fungi naturally make antibiotics, they're never in concentrations high enough to kill the surrounding cells. It's very, very low concentrations. Not strong enough to kill any surrounding cells. So what they're used for is communication. When bacteria or fungi naturally make antibiotics, they use them to communicate with other cells. You might recall quorum sensing in biofilms and other type of communication between cells. So naturally, antibiotics are used as molecules that allow cells to communicate with each other. Their antimicrobial action is coincidental. Turns out that they're normally used in communication, but in high enough concentrations, they kill. Okay? Just like you take too much of a medicine, it's going to be a bad thing. All right? So their antimicrobial action, they're not used to kill cells when they're produced by bacteria or fungi, but in high enough concentrations, they do. Okay, and that's what we want to make. We want to have them in a strong enough concentration where they can target those cells. Now, what about mechanisms of antimicrobial action? One of the real important things to think about is what we call selective toxicity. The drug or antibiotic or antimicrobial, whatever you want to call it, that drug, that agent has to be more toxic to the bacteria or virus or fungi or protozoan, whatever, but we'll just keep it to bacteria for simplicity here. Your drug, that has to be more toxic to the bacteria than it is to you, to the patient. Okay? Otherwise, you just defeat the purpose. All right? Now, the toxicity of any drug is dependent upon differences between the target and the host. The more differences there are between the target, the microbe, and the host, the easier it is to make a drug that targets the microbe specifically. So the more differences there are, it's easier for the host to avoid bad side effects or bad effects of that drug. So you want to think magic bullet, something that targets the microbe while not targeting the patient. And to do that, it's better if there's more differences between the target and the host. So, for example, here's a question. Why is it easier to find useful, in other words, low toxicity drugs against bacteria than it is to find them against fungi and protozoans? Any thoughts? Well, let's think about it. Remember, bacteria are prokaryotes. They're in their own domain. Fungi and protozoans and us are all eukaryotes. All right, so we have more in common. Our cells are more similar to fungi and protozoa. Our cells are much different from bacteria. So because our cells, eukaryotic, and bacterial cells, prokaryotic, are very, very different, it's easier to find a drug that's going to target just that bacteria and not our cells. Okay? What are two bacterial structures that the drug can target that eukaryotes don't have? Well, think about a number of things here. Remember, bacteria have their own type of ribosomes. They have peptidoglycan in their cell wall. Okay, nothing else has that. We can think of others here. All right? So, how do drugs work? Like, how do they kill or slow down the bacteria or fungi or protozoan or virus? Different drugs target different things. All right, so some drugs inhibit cell wall synthesis or break down the cell wall. Antibiotics often work by punching holes in the cell wall of the bacteria. Okay, they can inhibit or break down DNA or RNA, thereby slowing the bacterial cell down or destroying it. They can inhibit or denature proteins. They can disrupt the plasma membrane. They can inhibit or slow down metabolism. Or they could prevent the pathogen from attaching and then entering the cell, especially if it's a virus. And a drug can do more than one of these, although most drugs typically focus on one of these things here, on how they're going to target that microbe. So you can see here, we might inhibit the pathogen's attachment or entry. Here are some drugs that work that way. Okay? We can inhibit DNA or RNA synthesis. Here's some others that do that. Inhibit metabolism. Disrupt the plasma membrane. All right? Inhibit protein synthesis, slow that down. And inhibit the cell wall or work against the cell wall. Okay? So these are all ways that we can target a particular microbe. Okay? All right, so I'll tell you what, everyone, that's a good place to end part 1 here. So I will see you momentarily for part 2 of this chapter on antimicrobial treatment. Okay, see you soon.